Ever wonder how doctors detect blood disorders like anemia or malaria? It all starts with analyzing red blood cells, those tiny, disc-shaped carriers of oxygen in our blood. But traditional methods, they're time-consuming and prone to human error. That's where RedDino steps in, a groundbreaking AI model that's rewriting the rules of blood cell analysis. So what makes RedDino special? Think of it as a supercharged microscope powered by machine learning. Unlike older systems that struggle with blurry images or rare cell shapes, RedDino uses advanced algorithms to accurately identify and classify red blood cells, even in tricky cases. This isn't just a small improvement, it's a leap forward in automated diagnostics. Here's how it works. RedDino builds on decades of research in AI and medical imaging. It's trained using massive datasets of blood samples, learning to spot patterns no human could catch. But here's the kicker, it doesn't just guess, it adapts. By leveraging techniques like linear probing and KNN evaluation, RedDino fine-tunes its predictions to fit real-world data, making it super reliable. Let's dive into the technical details. Most AI models rely on labeled data, but RedDino uses self-supervised learning, a method where it learns from raw data without explicit labels. Imagine teaching a child to recognize cats by showing them thousands of images and letting them figure out patterns on their own. RedDino does something similar, but with blood cells. The team behind RedDino didn't just slap a generic AI model onto RBC analysis. They fine-tuned a framework called DINOV2, optimizing it for the unique challenges of red blood cells. Here's the twist. RBCs are remarkably uniform in shape and color, which can trip up standard AI. So the researchers tweaked the model by removing a regularizer that's usually helpful for natural images, because it was overprotecting RBCs, making them blend together. They also replaced some image adjustments with custom tweaks, like using 32-pixel level changes instead of pixel-by-pixel ones. The result? A model that sees the subtle differences between healthy and abnormal cells. The proof is in the results. RedDino outperforms existing models in classifying RBC shapes, a task critical for diagnosing conditions like sickle cell anemia or malaria. In tests, it beat the competition by a significant margin. Think of it as a chef who not only follows a recipe, but invents a better one. But here's what really sets RedDino apart. It's not just good at one task. The model generalizes across different imaging setups, staining techniques, and even patient populations. That's a big deal because real-world medical data is messy and varied. Whether it's a blood smear from a lab in Italy or a sample from a mobile clinic in Africa, RedDino adapts. How did they measure success? They didn't just look at plain accuracy. They used balanced accuracy, which matters when some cell types are rare, and weighted F1 scores, which balance precision and recall. For example, on the Chula and DSE datasets, both tricky because of imbalanced classes, RedDino still dominated, except in one metric where it tied. That's impressive, especially since Chula was part of the training data for other models. The real magic. Visualization. Using tools like PCA and UMAP, the researchers showed how RedDino's features separate abnormal cells in a 3D space. For instance, malaria-infected cells, highlighted in red and echinocytes, spiky cells, formed distinct clusters, proving the model's ability to distinguish subtle anomalies. Now let's talk about the numbers. The researchers tested RedDino against models like RaySnet 50 and DINOV2 on a dataset called Elsafty, which has 4 distinct data sources, like 4 different labs. They used a leave one source out approach, training on 3 sources and testing on the 4th, then rotating through all combinations. Why? Because in real-world clinical trials, data can vary wildly due to equipment, protocols, or even the person handling the samples. The goal? To see if RedDino can stay accurate despite these batch effects. And the results? RedDino blew the competition away. In linear probing, it was over 2.1% better than the next best model. For KNN tests, the gap was even bigger, 3% improvement. These aren't tiny tweaks, they're game changers. Imagine a model that's not just smart, but robust enough to work across different labs without needing a complete retrain. But how did they achieve this? RedDino's architecture is key. It's trained on patched smear images, not individual cells. Think of it like learning a puzzle by studying whole pieces instead of single tiles. It gives the model a better sense of context. And the RedDino base model? It's a powerhouse, balancing performance with efficiency. With 86 million parameters versus 304 million for others, it's like a high-performance engine that doesn't burn extra fuel. This isn't just about better tech, it's about saving lives. With RedDino, doctors could diagnose conditions faster, personalize treatments, and even catch diseases in their earliest stages. The next time you think about blood tests, remember, behind the scenes, AI like RedDino is working hard to make healthcare smarter, quicker, and more precise. Stay tuned for the next part, where we'll dive into how this tech is already changing labs around the world.